Okay, so welcome to this next video in which we are discussing defects in excitation contraction coupling in hypertrophied cardiomyocytes. Okay, so we've discussed that when the heart is initially struggling, okay, so when, for instance, the left ventricle is struggling to pump blood from the left ventricle chamber into the aorta, then what that leads to is pathological remodeling of the cardiomyocytes within the ventricular wall. Okay, so what happens is the cardiomyocytes undergo concentric hypertrophy, where their bellies thicken in width hugely, but they don't increase much in length. Okay, so this initially means that the cardiomyocyte is capable of generating a greater amount of force when it contracts. Okay, so initially its function goes up, so initially it's helpful to have pathological remodeling of the cardiomyocytes. However, gradually what will happen is the function of this hypertrophied cardiomyocyte will just decline and decline and decline, and its ability to generate force goes well below the ability of the initial unhypertrophied cardiomyocyte to generate force. Okay, and what gradually happens is as more and more of the left ventricular wall becomes these hypertrophied cardiomyocytes, then the ability of the left ventricular to contract and generate force just gets lower and lower and lower until eventually the cardiac output, the amount of blood that the left ventricle is ejecting in a minute into the aorta, gets so low that it's unsatisfactory. Basically, it's not enough now to meet the needs of the body, so the heart is failing basically, and that's known as heart failure or cardiac failure, when the heart isn't pumping, isn't strong enough to pump enough blood around the body in a minute to meet the needs of the peripheral tissue. That's when we say you are in heart failure, and it's a very serious condition which usually leads to death. Okay, right, or the need for a heart transplant. Right, so if you're lucky enough to get a heart transplant. Okay, right, so we are now investigating why these pathologically hypertrophied cardiomyocytes, which have this much bigger, thicker belly and not a hugely increased length, why these begin to reduce as far as their level of force that they are capable of producing is concerned. So why do they undergo this drop in their ability to contract, basically? Well, what we know is that if you look at these cardiomyocytes, basically when this hypertrophied cardiomyocyte undergoes an action potential, okay, so when it's stimulated to contract, the calcium signal that you see within the cytoplasm of the cell, the rise in calcium that occurs within the cytoplasm of the cell, known as the calcium tide, that is lower in these hypertrophied cardiomyocytes, okay? Now, what we also see is that the calcium sparks that are occurring in this cardiomyocyte, basically you see calcium sparks occurring even before an action potential has actually arrived in this cardiomyocyte. Now that is not what is supposed to happen. What is supposed to happen is that the action potential arrives at this cardiomyocyte, let's say this normal one here, and that causes the opening of the L-type voltage-gated calcium channels all over the cell, which causes loads of calcium sparklets, which diffuse across the many dyadic clefts for the many calcium synapses in the cell, and that calcium binds to the type 2 ranitidine receptors on the sarcoplasmic reticulum membrane, and these type 2 ranitidine receptors then open, releasing calcium from the SR, causing this calcium spark, and then the summation of all the calcium sparks everywhere for all of the calcium synapses is what then causes the global, i.e. whole cell, rise in calcium level in the cytoplasm, known as the calcium tide. I.e., you are not meant to get these calcium sparks before the calcium synapses are actually activated, and the calcium synapses only become activated when the cell undergoes an action potential. So, basically in these pathologically hypertrophied cardiomyocytes, you see calcium sparks occurring all over the shot in this cell, long before an action potential has ever occurred. So we're now going to investigate why this occurs. Now, in another video, what we have talked already about is how in these pathologically hypertrophied cardiomyocytes, the SR luminal store of calcium is depleted. So the amount of calcium stored in this essential organelle within the cell is lower in these hypertrophied cells, and this is because of the decreased function of the SERCA2 pump, which pumps calcium into the calcium lumen. But we've got many different videos on that. Okay, so we won't talk about that now, but that's one of the reasons the calcium tide goes down, because if you've got less calcium in the lumen of the SR, then the amount of calcium you can release in the calcium spark is lower, and since the calcium tide is made up of the sum of all the calcium sparks, it also contributes the calcium sparklets also contribute a tiny bit, but they're trivial compared to the calcium sparks. Their main function is in causing the calcium sparks. Okay, so if the calcium sparks are all smaller, the calcium tide is going to be smaller as well. So that's one of the contributing factors to this, but let's now look at the actual factors within the excitation contraction coupling which contribute to the reduced calcium tide. Okay, right, so, well, basically you could have defects, in all of this machinery here, and that would lead to lower calcium sparks. So, for instance, if the L-type voltage-gated calcium channels have reduced conductance, okay, then when they open, they'd let in less calcium, so the calcium spark that would be smaller, and this would then activate fewer type 2 ranitidine receptors. So although I've only drawn it activating one, of course there'll be many type 2 ranitidine receptors on this process of the SR, and this calcium spark that needs to activate as many as possible. So if the calcium spark that is smaller, then it stands to reason that it will activate fewer, and therefore the number of type 2 ranitidine receptors opening over here on this SR membrane here will be lower, so the size of the calcium spark produced will also be lower. Now, what we do not find is we do not find defects in the L-type voltage-gated calcium channel, as I've just described, in pathologically hypertrophied cardiomyocytes. So this does not seem to have any problem with it. So we can rule out the L-type voltage-gated calcium channel. It's functioning absolutely fine. Okay, now let's move on to the structure of these calcium synapses. Okay, so what if there was just problems with the actual calcium synapses themselves? So maybe, for example, the L-type voltage-gated calcium channels aren't positioned near the type 2 ranitidine receptors. Okay, so let me show this basically on the next page. So, for instance, what we could have is a mismatch. Okay, so let me show this. We can have a mismatch, i.e. the calcium synapses just are not formed properly. So if we have our T-tubule here, then let's say the L-type voltage-gated calcium channel is over here, but the process from the SR is here, okay? And that means the type 2 ranitidine receptors are here. So as you can see now, you haven't got the L-type voltage-gated calcium channels, which I'll draw in this vivid purple colour here. You haven't got the L-type voltage-gated calcium channels anywhere near the type 2 ranitidine receptors, which are over here in turquoise. So this is what's known as a mismatch, basically. You're not going to have a functional calcium synapse now, because when the action potential comes along, these L-type voltage-gated calcium channels are going to open. They're going to allow in calcium, the so-called calcium sparklet, but this calcium isn't anywhere near the type 2 ranitidine receptors, so it's not going to activate the release of calcium from the SR, so all you're going to get basically is the calcium sparklet now, with no calcium spark. And the calcium sparklet is trivial, basically. It's a tiny little amount of calcium. It's not going to contribute that much towards the calcium tide at all. So this would explain, then, why the calcium tide in these hypertrophied cells was lower. Okay? Now, we don't see this happening, so this is another thing that does not seem to happen in cardiomyocytes. Now, let's try and think of other defects that could potentially be there in the structure of the calcium synapses. Well, another defect that could be there, and this time this is going to be one that we actually do see, is there could be just a too big gap. So the size of the dyadic cleft could simply be too big. So again, here's the L-type voltage-gated calcium channel, and this time it's in the right position, i.e. it's interfaced with the the type 2 ranitidine receptor on these sarcoplasmic reticulum processes. But now, the size of the dyadic cleft, the gap between the plasma membrane and the SR lumen, is just too big, okay? So, even though the action potential comes along and activates the L-type voltage-gated calcium channel here, or the dihydropyridine receptor if you prefer that name. So this is going to open, release this, allow this calcium to enter the cell. So you will get this calcium sparklet, but now too little of it is actually going to make it across the dyadic cleft to activate that many type 2 ranitidine receptors at all. So you're not going to get as much activation of the type 2 ranitidine receptors, which means that the amount of calcium you're going to get released from the sarcoplasmic reticulum is going to be reduced. So the size of the calcium spark is going to go down. So here's the calcium sparklet. And now what's going to happen is the size of the calcium spark, which remember is the name for the calcium signal produced by the opening of the type 2 ranitidine receptors, it's going to be much reduced basically. And this is in accordance with what we see in cardiomyocytes. We see that the individual calcium sparks are actually smaller. So basically this actually does occur in cardiomyocytes that have undergone pathological hypertrophy. At least we think it does. So you do find increased sized dyadic clefts within the calcium synapses of hypertrophied cardiomyocytes. So this one is something that happens, and this is something else that contributes to the reduced calcium tide within cardiomyocytes. Okay, now let's discuss one other problem that could occur with the calcium synapses then, before we move on to the type 2 ranitidine receptor. Well, basically what you could get is you could just get atrophy of the T-tubules, okay? So basically, what if the T-tubules were just invaginating into the cytoplasm far too shallowly, okay? So then effectively, a way of gaining some intuition for what the T-tubules are is to imagine a balloon into which you've stuck your fingers, you've invaginated your fingers. That's very similar to what a T-tubule is, effectively. The cell membrane is like the surface of the balloon, and T-tubules are effectively finger-shaped invaginations of the surface of the balloon, okay? So imagine now that they're just not that deep. You're only maybe sticking in the distal phalanx of the finger, basically, this portion here, not the entire thing. So that would make them very shallow, okay? Then these processes from the sarcoplasmic reticulum, they'd still be there, but they've got no T-tubule to actually synapse with, okay? So this is what's known as T-tubule atrophy. Okay, so atrophy just means sort of wasting away. Okay, so as you get older, you get muscle atrophy, or muscle just wastes away. Right, okay, so these calcium synapses down here now, they're completely useless, because the type 2 ranitidine receptor is sitting here, but it's not juxtaposed with the plasma membrane anymore. It's not connected with the L-type voltage-gated calcium channel, so it's not going to receive any calcium sparklet, which is telling it to open. So these processes of the SR that are down here, which no longer have the T-tubule, those are totally useless now, so they're not going to contribute anything. So overall now, you're going to get fewer calcium sparks happening because you've got fewer calcium synapses that are actually active. Okay, so that's going to mean that overall the calcium tide, which remember is the sum of all the calcium sparklets, is also going to be reduced. And T-tubule atrophy is also seen in hypertrophied cardiomyocytes. Okay, so we've seen two things that contribute then to the reduced size of the calcium tide within cardiomyocytes. We've seen an increased gap, an increased diatic cleft. We've seen increased two-tubule atrophy. Okay, we've also seen the circa pump, the problem with the circa 2 pump, which leads to less calcium in the SR lumen. So that also means that the release of calcium in the form of the calcium sparks is lower. These are all contributing to the calcium tide being smaller. Now, if the calcium tide is too small, then the number of sarcomeres within the cardiomyocyte that you are actually going to activate to start contracting is going to be fewer. So basically, when you actually cause a calcium tide, you don't make absolutely every single sarcomere contract to its full potential. If the calcium tide is bigger, then you're going to recruit more sarcomeres to the actual contraction process. So the number of sarcomeres which will actually be undergoing contraction to their full potential is going to be greater, so the force you'll produce is greater. So if calcium tide goes down, that means that the number of sarcomeres that you're going to activate contraction in is going to be lower, and therefore the force that you generate is also going to be lower. So this could underlie why these hypertrophied cardiomyocytes have a lower force of contraction. In the next video, what we'll talk about is why you get these sporadic calcium sparks in hypertrophied cardiomyocytes.